Apicomplexa of systemic pathology in Toxoplasma. The outline of this presentation: first is the introduction, second, the morphology, the terminology review as it relates to Toxoplasma Gandhi, the life cycle rather complex, the pathogenesis, the clinical disease, the diagnosis, the treatment, especially in cats, the control, non-valid paratenic host, and zoonosis in human infections. Toxoplasma Gandhi is an obligate intracellular parasinic protozoan that causes toxoplasmosis. It is found worldwide. It is capable of infecting virtually all warm-blooded animals but felids, such as domestic cats. It is the only known definitive host in which the parasite may undergo sexual reproduction. The morphology of Toxoplasma Gandhi: it is an intracellular parasite of intestinal epithelium and various host tissues. The oocyst: it is a small, subspherical, smooth coat, no polar cup, and single embryo when passed. Sporulated oocyst contains 4 sporocysts with 2 sporozotes each. 8 sporozotes in total. For the terminology review as it relates to Toxoplasma Gandhi: the definitive host: it is a species in which the adult or sexual form of the parasite occurs. Felids are definitive host for Toxoplasma Gandhi. Facultative intermediate or paratenic host: it is a species which supports the multiplication or asexual stage of a parasite. It can carry parasites through definitive host but are not required for life cycle of the parasite. It is any warm-blooded animal. Accidental host or dead-end host: parasites are not passed on and can complete life cycle. Humans are accidental host when infected with Toxoplasma Gandhi. Endogeny: it is a single division forming 2 daughter cells inside cytoplasm of a mother cell. It occurs in the cat and paratenic host, the only replicative form in paratenic host. Schizogeny: it is a multicellular division followed by a cytoplasmic division forming multiple daughter cells. It occurs only in the cat intestinal epithelium. Toxozoa: it is a fast-growing infective stage. It is localized in neural and muscle cells. Fast-growing, will develop into bradyzotes. Bradyzote: it is a slow-growing infective stage. It is localized as cyst in tissue, especially in neural and skeletal and remain semi-dormant in chronic infections. Life cycle rather complex in Toxoplasma Gandhi. First, in cat to cat. The direct life cycle: homogeneous. The definitive host only in felids. Next, the transmission: focal oral. Ingestion of oocyst only in felid host. The invasion: the sporozotes exist from oocyst and invade enterocyte. Some sporozotes invade deep tissue, the CNS, muscle, and viscera, multiply and disseminate as satyzotes, eventually forming bradyzote cyst. Asexual reproduction in intestinal cells. First, the endogeny and schizogeny. It is approximately 5 cycles of endogeny and schizogeny. It causes none to mild pathology in the cat. Sexual reproduction: it only occurs in the felids. Final generation of zotes exit the enterocyte. It infects other enterocytes and go through gametogeny, the production of gametes. Macrogamete: some final zotes remain a single cell and become a macrogamete within a macrogamete. Microgametes: other final zotes go through multinuclear division, cytoplasmic division, and develop 2 flagella, or biflagellate on each gamete, thus forming a macrogamete. Exflagellation: it is when microgametes exit the microgamete in search of a microgamete. Next is the fertilization. A microgamete fuses with a macrogamete, forming a zygote. Next, a cyst wall forms around the zygote, and the immature oocyst exits the microgamete into the lumen of the host gut and is passed in the feces. Next is the dissemination. Oocysts, the unsporulated, exit the host in the feces and contaminate the environment. First, felids are the only host to pass to oocysts because of sexual fertilization. Second, prepatent period. Oocyst ingestion to oocyst passing 19 to 48 days. Third, oocysts highly resistant and remain infectious for many months. The paratenic host to cat. It is a facultative, indirect life cycle or the heterogenous. Paratenic host. Any warm-blooded animal infected by ingesting sporulated oocysts from infected cat feces. Next is the transmission to cat. Cat ingest of a tissue cyst from a paratenic host. Carnivorism. Next, bradyzotes released from tissue cysts develop into toxicotes. Next, the invasion, asexual reproduction, intestinal epithelium, sexual reproduction, and sporulated oocysts. Some sporozotes invade deep tissue. Next, the dissemination. Unsporulated oocysts pass in feces. Sporulate in environment. Lastly, the prepatent period. The ingest tissue cyst from paratenic host to passing oocyst. 3 to 10 days versus the 19 to 48 days in the cat-to-cat cycle. To paratenic host. In facultative indirect life cycle, it is heterogeneous to paratenic host in any warm-blooded animal, including felids. The transmission to paratenic host. The ingestion of an oocyst from the feces of a felid, the definitive host. Next, the invasion. The zotes enter intestinal cells and lymph cells and transform into toxicotes. Zotes, rapidly dividing zotes via endogeny, the two daughter cells or division. Asexual reproduction, the extra-intestinal cells. The tachyzotes are dispersed throughout the body of the paratenic host. The rapid destruction of host cells during the toxicote phase causes the acute severe disease. Eventually, toxicotes transform into bradyzotes. Bradyzotes, slowly dividing zotes via endogeny, the two daughter cells or division. Next, the stationary, non-disseminating and form tissue cyst. The tissue cyst cause more chronic pathology in the paratenic and human host. It occurs in and cause damage to the brain, liver, striated muscles, and remain viable for the life of the paratenic host. General notes. Tachyzotes may be transplacentally transmitted. Thus, mother ingestion of oocysts from cat feces, toxoplasmosis in fetus. Paratenic host to paratenic host. First, the transmission. The ingestion of a tissue cyst from a paratenic host in any warm-blooded animals. Carnivorism. Next is the invasion and asexual reproduction is the same as cat-to-cat paratenic host. General notes. Tachyzotes may be transplacentally transmitted. Thus, mother ingestion of raw meat with tissue cyst, the toxoplasmosis in fetus. For the pathogenesis, intestinal phase in felids, minimal. Systemic disease, The extraintestinal phase in felids, paratenic host or humans, explosive. Multiplication of tachyzotes cause massive destruction of host cells, also acute immune response. Tissue cysts cause tissue damage and a source for recrudescence of acute disease. The clinical disease, only in cat. The intestinal disease, usually no complaint. Cyst found on routine focal. Some cats develop self-limiting small howeldaria. The systemic disease, pneumonia, the main sign in feline toxoplasmosis, especially in FIV plus cats. The fever, anorexia, vomiting, diarrhea, myositis, uveitis, enlarged lymph nodes, encephalitis, and nephritis, and death. Diagnosis: the intestinal disease, oocyst in feces, only in cats. Use zinc sulfate as oocysts may disperse solutions. In systemic disease, serogenic test good to rule out toxoplasmosis if seronegative. Because so many are exposed to Toxoplasma, a seropositive result doesn't necessarily infection or cause of the cat's disease. It could be incidental finding. Treatment, only in cat. Pyrimethamine plus triple fulva drugs used against intestinal phase and acute phase. Also, clindamycin and ponazuril is used. Clindamycin is used in systemic disease. No good treatment for tissue cyst.
Control, especially in cat. Sanitation. Clean litter box daily. Sporulation in 1 to 3 days. No raw meat for cats. Lastly, don't let cat outside to defecate or hunt.
Felid paratenic host. The dogs, sheep and goats, cattle, horse, rodents, swine, and poultry. In dog, it is a systemic toxoplasmosis, ingest oocysts from cat feces or tissue cysts from prey. In sheep and goats, the systemic toxoplasmosis and congenital toxoplasmosis ingest oocysts from cat to cat feces. In cattle, congenital toxoplasmosis, the abortion but very rare, ingest oocysts from cat feces. In horse, systemic toxoplasmosis, low pathology, ingest oocysts from cat feces. In rodents, it is systemic toxoplasmosis, ingest oocysts from cat feces or tissue cysts from prey. In swine, systemic toxoplasmosis, ingest oocysts from cat feces or tissue cysts from prey. In poultry, also systemic toxoplasmosis, ingest oocysts from cat feces, prevalent in free-range and backyard chickens. It is important source of infections for humans. The zoonosis, systemic toxoplasmosis and congenital toxoplasmosis, ingestion of oocysts from cat feces, unwashed vegetables, or unclean hands. The ingestion of tissue cysts from undercooked meat, mainly mutton, goat, free-range pork, and free-range chicken. The transplacental transmission. The immunocompetent or systemic toxoplasmosis. First exposure is the flu-like illness that may last for weeks. The future exposures. The immune protected and it has no tissue. Immunodeficient adult. Elderly and those with immunosuppressive diseases. Severe disease. First exposure prior to immune suppression, then recrudescence may cause severe disease. First or future exposure after immune suppression may cause severe acute disease. For the immunodeficient fetus, the congenital toxoplasmosis, severe disease or congenital malformation, mental retardation, and death. If mother's first exposure occurs during pregnancy, transplacental transmission is greatest in third trimester, but more severe defects if transplacental transmission occurs in the first trimester. The first exposure prior to pregnancy, then immune system controls toxoplasmosis unless mother is immune deficient and can get antibody tested. Avoid cat feces. Have someone else clean litter box daily. Inspiration in 1 to 3 days. Avoid uncooked meat, unclean hands, unclean vegetables, unclean knives and cutting boards.